Press the power button to proceed.
What does this actually say? What is it?
Hello there, my name is Obot. I want to show you something today. Let us proceed. Let's also change the music.
Screening is mainly for the production of highly desirable strain. It means that it is a method to obtain our desirable strain. Isolation and detection of high-yielding species from the natural resources material containing a heterogeneous microbiome population. Among the characteristics of the strain production is it should be in high-yielding strain. Second, have stable biochemical characteristics. Third, not produce undesirable substances. And lastly, be easily cultivated on a large scale.
Now you know the definition. Let's go to the next one.
Now I will talk about the types of screening techniques for industrial microbes. There are many types of screening techniques, but today I will only focus on two techniques, which are primary and secondary screening. Firstly, primary screening may be defined as the detection and isolation of the desired microbes based on its qualitative ability to produce the desired product like antibiotic, amino acid, and enzyme. In this process, desired microbes is generally isolated from a natural environment like soil, which contains several different species. Sometimes, the desired microbes must be isolated from a large population of different species of microbes. Like I have mentioned before, the functions of primary screening are just for detection and isolation of microbes, but it cannot give the details of production potential or yield of the organism. Such details can be discovered by further experimentation and are known as secondary screening. Secondary screening can provide a broad range of information such as the ability of the organism to produce a metabolite that can be used as an industrial organism. Second, the quality of the yield product. Third, the type of fermentation process that can perform. And lastly, elimination of the organism, which are not industrially important. To evaluate the true potential of the isolated microbes, both qualitative and quantitative analyses are generally conducted.
Pretty informative, right? Next one.
There are many screening methods for industrial microbes, and in particular for primary screening, we have crowded plate technique and oxonography technique. Okay, so for crowded plate technique, there are pros and cons in using this. The advantages of using crowded plate technique is that it is the simplest technique for the detection of antibiotic producing microorganisms from soil samples, and it is also a fairly rapid, in which it only takes a couple of days to produce any results. But the disadvantages of using crowded plate techniques is that it only detects microorganisms which produces compounds to kill bacteria found in their immediate environment. These compounds can potentially be toxic to humans, and they may be lethal only to certain types of bacteria, such as soil bacteria, and it is opposed to the bacteria that can actually cause disease in humans. Moreover, they can only detect microorganisms that start to produce antibiotic compounds within a couple of days of being cultured and incubated. So this can miss other compounds that is potentially be of interest to the researchers who are using crowded plate techniques. The procedure of using this technique has two parts, the plating part and the purification part. For the plating part, first, a sample of organism from soil or any other sources is diluted in water and then spread onto petri dishes containing agar gel, which is rich in nutrients for the bacteria to grow. Scientists will then select plates that have large number of colonies and then look for microorganisms that have inhibited the growth of other microorganisms in their vicinity. These microbes are possibly secreting some kind of compound that can kill their neighbors. In the purification part, the colonies that are suspected to produce antibiotics are then transferred to another plate so that they can be purified and grown in isolation. It is entirely possible that the bacteria is just altering the pH of the environment, making changes that are not related to antibiotic production. Henceforth, further tests are needed to confirm that it is an antibiotic producing strain. Nonetheless, the crowd plate technique was sometimes helpful in identifying microorganisms that could serve as sources of new antibiotics. Another part of primary screening is oxonography, and according to Perkins, oxonography is a quick, economical, highly efficient method of determination of growth requirements of an auxotrophic mutant. Some advantages of using oxonography is that they have many growth factors that can be tested on a plate. The back mutations appear as discrete colonies. High concentration of growth factor. If it is toxic, it will not fail eliciting response. Double requirements may be identified by a zone of growth between two supplements. Inhibitions by other supplements are identified by the failure of spreading growth in the vicinity, and the substance difficult to sterilize can be tested. For the procedure of using oxonography is firstly place approximately 1 ml of conidial suspension in a minimal medium. When using non-conidiating strain, a suspension of macerated mycelial fragments can be used. Add the substances to be tested into a solidified agar and incubate at 34 degrees Celsius for 12 hours because some responses can be seen as little at that time. For testing solid substances, small quantity of the test mixture is placed on surface of agar using flame flattened platinum iridium blade as a microspatula and taking the substance direct from the reagent bottle. For liquids, a small droplet is delivered from a Pasteur pipette. Test substances does not need to be sterilized. If the strain being tested has an already known requirement or in addition to the unknown requirement, then the substance can be supplied by adding it to the minimal medium using for plating. And lastly, the requirements of unknown auxotrophs can be keyed out stepwise using 2 or 3 successive plates. All 3 may be prepared at the same time, and those not used in the first step are refrigerated until they are needed.
That's a lot to take in. Can you still keep up?
The enrichment culture technique is an isolation technique designed to make conditions of growth very suitable for organisms of interest, while having unsuitable environment for any competition. The purpose of this is to purify sample in order to obtain the organism of interest. Furthermore, it used to increase a small number of desired organisms to detectable levels. So how the enrichment culture technique execute? Let's imagine it together. You have a broth media contain select nutrient for the growth of desired organism. Put the sample in the broth media and leave it for the desired organism to grow and others to die. One way to know whether the enrichment culture technique is well executed: pour the culture onto the agar such as blood agar and leave it to grow so that the result can be observed to determine it. Indicator dye technique used to detecting microorganisms that able to produce organic acid. It use pH indicated dye such as neutral red, chromatic blue, and many more.
Very useful technique. Next one, please.
Now, let's talk about secondary screening. Secondary screening allows further sorting out of microorganisms obtained from primary screening, having real value for industrial processes and discarding of those lacking this potential. Secondary screening helps in predicting the approaches to be utilized in conducting further research on the microorganisms and its fermentation processes. Secondary screening should also reveal whether the microorganisms are able to chemically alter or even destroy their own fermentation products. Moreover, it should determine whether microorganisms are actually producing new chemical compounds that are not previously described. Secondary screening is conducted on agar plates in flasks or small fermenter containing liquid media. One of the purposes of secondary screening is it should give information about the evaluation of the true potential of the microorganisms for industrial usage. Next, it should show whether certain medium components are missing or possibly are toxic to the growth of the organisms or its ability to accumulate fermentation.
Can you memorize all 6 of them?
Medium selection. Under this topic, I would like to talk about 2 things, which are microbes growth requirements and medium ingredient in industry. The first one is microorganism growth requirements. There are 6 elements which are needed by microbes to grow and reproduce. Oxygen, as electron acceptor in respiration for aerobic microorganism. Mineral, act as trace elements. Water, all bacteria need moisture or water in a usable or available form to grow and reproduce. Chelators, to detoxify poisonous metal agents. Energy, microorganism can derive energy from source of carbohydrate, mostly glucose, alcohols, and amino acids. Vitamins, needed as coenzymes. Carbon and nitrogen, as main component of cellular material. The second one, typical medium ingredients that are required in industrial and also can be applied to the laboratory. There are 8 ingredients which must be included in media preparation in order to allow microbes to grow and reproduce. Number 1, carbon and nitrogen, as main component of cellular material, and nitrogen sources, for example NH3, component of amino acids, nucleic acid nucleotides, and coenzymes. Number 2, vitamins, for example vitamin K and B12, as microbial growth factors. Number 3, minerals, for example zinc and copper, as trace elements. Number 4, inducers, which regulate gene expression of microorganisms. Number 5, precursors, for better yield of desired products. Number 6, inhibitors, for example potassium metabisulfides (KMS), which limit growth of wild yeast and bacteria in beer medium. Number 7. Antifoams. For example, sulfosuccinates for defoaming composition. Last one, chelators to detoxify poisonous metal agents.
That one was longer than before. Hope you're still here.
Crude and defined media. Media can be assigned into two types, defined media and crude media, also known as complex media. Defined media, made from pure compounds, free of unknown biological activities such as enzyme and growth factors, which may affect the responses being studied. The effect of nutrient on growth and yield of product can also be analyzed. Crude media, made from complex mixtures, which is from agricultural product. Individual ingredients may supply more than one requirement, may contain polymers or even solids. Media for industrial bioprocess. Crude and defined media. Crude media is made up of unrefined agricultural product containing barley. Crude media is cheap, but composition is variable. Crude media is used for large volume in expensive product, such as biofuel from whey. Crude media contains high level of nutrient, vitamin, protein, growth factor, anti-foaming agent, and precursor. Defined media are like those we use in the lab, such as minimal salt mediums. Defined media is expensive, but composition and specific concentration is known and should not vary. Defined media is used from expensive low-volume products such as anti-cancer drugs. Here we look at defined media versus crude media. Defined media, also known as synthetic media or chemically defined media, while as crude media, also known as complex media or rich media. The definition of defined media are culture media that contain a known chemical composition, while as crude media are cultured media that contain an unknown chemical composition. The ingredient in defined media contain pure biochemical, while as in crude media, it's contain complex material of biological origin such as blood, milk, and yeast extract. Defined media only provide exact nutrient needed by the organism for growth, while as crude media contain full range of growth factor that may be required by an organism. An example of defined media are glucose salt broth, while as an example of crude media are nutrient agar, tryptic soy broth, or tryptic soy agar.
It's really important to know about our media too, right?
There are various criteria to be considered when preparing a medium. I have listed these criteria into three major categories as seen on the slideshow. The first major criteria that I have listed is product yield. Obviously, the main point of these industrial microbes is to yield product. A proficient media should minimize the use of substrate while also maximizing products being produced. Though we want to maximize the product yield, we should also minimize the undesired products, also known as the byproducts. A simple example for this criteria is the fermentation process. The fermentation process utilizes sugar as a substrate, and the products produced are ethanol and carbon dioxide. A proficient media should minimize the use of the sugar while also maximizing the production of ethanol, but also minimize the byproduct carbon dioxide. Another criteria that should be taken into account is the availability and quality of the media. The media or the materials needed to prepare the media should always be available throughout the year. This is especially true in the industrial sector as they need to always be prepared to meet consumer demand. Quality of medium is crucial and it should always be consistent. The quality of the media depends directly on the quality of the raw materials used for their preparation. An example is water. Water is examined for presence of copper ion. Ideal water should not contain any copper ions as these ions inhibit growth of microorganisms. To maintain the consistency of the quality, proper quality management system is essential. Media preparation should be quick and efficient while also being cost effective. Avoid using media that may cause issues in other processes as it will slow down production. Take note of the media's sterilizing methods and ensure it is not too tedious or too expensive.
Medium preparation. It is a crucial process as the fermentation medium could affect resultant product concentration, yield, volumetric and fermenter design. Theoretically, less optimum media produce less product, while optimum media produce maximum product. Medium utilizing anaerobic microbes doesn't need fermenter conditions that support aerobic growth, such as impeller and sparger to improve aeration. Like other living organisms, it is essential for microbes to obtain macronutrients and micronutrients. Media formulated for microbial growth usually requires carbon, nitrogen, growth factors, minerals, and chelating agent. For medium design, we need to consider about medium effect on strain, aperture selection, scale-up aspect, and ability to support metabolic activity. For example, to culture aerobic non-lactose fermenter bacteria, we shouldn't use media with lactose and should use buffer alanine to promote optimal oxygen transfer. It also contains essential amino acids and properly redesigned to scale up, such as increase the substrate concentration to increase output products.
Hope you enjoyed that. Thank you and see you again.